never alone, even in the confines of our own bodies. We're hosts to trillions of microorganisms. Our cells are outnumbered by bacteria. It's like finding out you're not the lead actor in your own movie, but just someone with a walk-on role. The complex population of microorganisms we carry around is uniquely ours, and deeply involved in the plot of our health story. And maybe it has been steering the ship all along. I'm Silvia Lazzaris, and this is The Science of You, a podcast series about the groundbreaking science that's changing the way we approach healthcare. Together with those who are leading the charge, we will journey from the discoveries that transformed our understanding of the human body to the innovations that promise to revolutionize treatments and medical practices. The Science of You is a Cora Media podcast, supported by the European Partnership for Personalized Medicine, coordinated by Fondazione Toscana Life Sciences, and co-funded by the European Union's Horizon Europe Research and Innovation Programme.
The more that we looked, the more we saw that there's, this huge diversity of microbes, particularly in our guts. And then the question, of course...
This is Emma Allen-Vercoe, professor of microbiology at the University of Guelph in Canada, and one of our guides throughout this episode. For most of human history, we didn't know what caused infections. People blamed bad air, bodily humors, divine punishment, or maybe a curse from the local witch. Rituals and prayers were our go-to remedies. But diseases like smallpox and cholera kept wiping us out until we realized our bodies aren't just our own. They're home to trillions of microbes. Some bad, but many essential to keeping us healthy.
Well, I think actually the aha moment wasn't really a moment. It was kind of a slow realization. So we've always known that microbes have been living on us and in us, even from the time of, way back to Antony van Leeuwenhoek. He was the pioneer of microscopy, and he showed that...
the guy she's talking about, Dr. van Leeuwenhoek, was a self-taught Dutch scientist who back in 1681 decided, in a moment of curiosity or maybe desperation, to put his own watery stools under a microscope he had built himself. He was the first to see these tiny creatures, which he called, endearingly, animalcules. Despite his discovery, it took centuries for scientists to connect microbes to diseases and health. Many scientists for centuries kept thinking that these minuscule organisms couldn't possibly affect the human body. But science, being science, thrives on questions and skepticism. So by the end of the 1800s, the germ theory of disease had finally taken root in the medical world. And, hallelujah, people actually started washing their hands. But most of the attention was on the microorganisms that made us sick.
There are a lot of microbes that cause disease, and those are the ones that obviously medical science is much more interested in. But as a result of that, the bias for study of microbiology for the past 200 or so years has been on the microbes which do damage, and in terms of human health anyway. And the microbes.
It was Ilya Mechnikov who came up with a revolutionary idea. A scientist born at the end of the 1800s in Kharkiv, at that time part of the Russian Empire, today a Ukrainian city. He was a man so dedicated to his work that he tried to kill himself twice, just for science. His idea was simple. Maybe, just maybe, not all bacteria are bad for us.
So the next question is, well, what do they do for us? And I think some surprising realizations have come out of that, sort of focusing on microbes instead of as pathogens, as sort of health-promoting organisms is a whole new way of looking at things. And when you look at things through a new lens, you see lots of new ideas and new thoughts come out. And I think that the biggest success story is in the treatment of a disease called Clostridioides difficile infection, or C. diff as it's commonly known.
The first real victory, in line with Mechnikov's thinking that good bacteria could help us fight bad ones, came in 1958 in Colorado, where doctors successfully treated 4 patients suffering from an infection called Clostridium difficile, also called C. diff, using someone else's feces.
It's a microbe that actually commonly lives in many of us. It has some very particular toxins that it makes under certain conditions. Now, when it makes those toxins, it makes them when it finds itself in a position that it can grow very easily, kind of unchecked. And that happens when you take an antibiotic. And so if you have C. diff or you contract C. diff when you're taking an antibiotic, you're at great risk of that C. diff being able to sort of multiply in your gut and to make its toxins. And the toxins actually are very, very dangerous to the gut lining. They cause ulceration and terrible diarrhea, very, very painful as well. What's happened there is that the antibiotic has wiped out or removed quite a large proportion of the gut microbiome, has allowed the C. diff to proliferate. So this is a disease caused by antibiotics is now treated with antibiotics. And you can imagine that for many people, this doesn't necessarily work very well.
But as soon as you remove the antibiotics, C. diff comes roaring back again. In desperation, a few bold doctors decided to try fecal transplants, essentially transferring healthy poop into a sick patient's gut. And remarkably, for C. diff cases, it works almost every time. In 2013, a trial trying to treat C. difficile was cut short. Not because it was failing, but because the results were so staggering with fecal transplants that the researchers didn't want to keep patients on the standard antibiotic treatment any longer. But there was another hurdle to overcome. Traditional microbiology had long been obsessed with studying individual species of microbes, as if they were lonely little islands, each in its own isolated unit. But these microbes don't grow and thrive easily in a vacuum. They flourish in complex ecosystems like our bodies, interacting with one another and with us. Because, in a way, much like the Earth contains a variety of different ecosystems, from rainforests to deserts, a single animal is full of ecosystems too. Skin, mouths, guts, genitals, any organ that connects with the outside world has its own community of microbes. That's why the introduction of the term microbiome in 2001 was so important. It marked a shift in focus.
Technically, it's the collection of bacteria, archaeobacteria, fungi, and viruses that are present in multiple human body sites. Most notably, maybe the gut, all the gastrointestinal tract, actually, the oral cavity, the skin, the urogenital tract.
This is Nicola Segata, professor of metagenomics at the University of Trento and at the European Institute of Oncology in Milan.
And these are complex and ecological entities. So in all these body sites, there are hundreds, if not thousands, of different species of these microorganisms that are interacting between themselves and with our body, with the so-called host.
So we've got the bacteria, which most people sort of think about when they think about microbes. There's also viruses, believe it or not. These are viruses that actually infect bacteria for the most part, although there are some viruses in there which affect humans. There's also yeasts and fungi. And we also have some really unusual microbes that we call archaea.
The understanding of these complex relationships was the goal of the Human Microbiome Project in 2012. What made the project really stand out was its focus not just on the microbiomes of sick people, but of healthy individuals too. To our astonishment, we discovered that our own cells are outnumbered by bacteria.
The ratio of the human cells to microbial cells was about 1 human cell to every 1.3 microbial cells. So we are actually more microbial than we are human, which is pretty astounding, I think, to most people. And you might think that that's icky, but it isn't icky at all, because a lot of those microbes do some very, very important work for us, and they help to keep us healthy, and all sorts of other interesting things.
Scientists also found out that the microbes in our bodies are as unique as our fingerprints. Every time we walk, talk, touch something, or sneeze, we unleash a personalized clouds of microorganisms into space. On average, each person aerosolizes around 37 million bacteria per hour. If we had microscopic view glasses, we'd see that we all walk through the world carrying a living aura around us. again, Nicola Segata.
If you take two random individuals in the world, they have on average 20, 30, 35% of species in common. So we studied, for example, couples living together or even students living in the same house. And we found out that at the beginning, they have nothing in common, 0.something% of things in common. But after 2-3 years, they arrive to 10, 15, 20% of strains that are exactly the same. People living together are sharing their microbiome.
While 20% might sound significant, flip the perspective, and this means that a full 80% of our microbiome is very personal. We're all walking around with our own little universe inside us. Let's take this further. Even if two people might have the same genetic predisposition to a disease, how it shows up could be vastly different depending on what's going on with their microbiome. The potential here is enormous. One day, doctors could predict more accurately who's going to get sick and how to treat them.
Gerald Hannett coined the term poo print. The idea here is that you could actually take a room full of people and you could take their stool samples and sequence them. And a year later, you could have the same people provide stool samples, which are anonymized. And once you sequence them, you could probably be able to match up whose poop is whose based on the profile that you get. So no one has actually done that experiment yet, as far as I can tell. But that's the principle that is there. They are so unique and individual that you could actually use them as an identifying feature.
Take a moment to consider how we might appear to these populations of microorganisms, the very architects of life on Earth. They've been around for billions of years. They've inhabited dinosaurs and mammoths. And now they find themselves squeezed into these tiny human bodies. With all that knowledge accumulated over geological eras, they've adapted in ways we can't even fathom. It's in their best interest to keep us functioning. But really, what do they do for us?
Humans are a real blip on the radar in comparison to how long microbes have been around. They're definitely very important in education of our immune system, making our immune system work optimally for us, and basically there to show us what's what is harmful and what is not. They are very good, obviously, at processing the food that we eat. I'm talking here specifically about the microbes in the gut now. And so you've probably heard of something called butyrate, which is a short-chain fatty acid. Everyone talks about butyrate now. It's a beneficial component of the metabolome that is produced by the gut microbiota.
Honestly, I hadn't heard of butyrate, so I looked it up. It's a short-chain fatty acid, and it's the prime energy source for the cells lining our colon. It also works as an anti-inflammatory and plays a role in keeping our intestines together. It's also been linked to the health of our metabolism. But our bodies don't produce it. Gut bacteria do, when they process fibers in our colon. It's what scientists call postbiotics, beneficial compounds that bacteria produce after breaking down food or other substances in our gut.
And so our microbes are feeding us with energy. And so the cells that line our guts are actually using that butyrate as an energy source, which I think is fascinating and sort of shows that co-evolution.
But that's surely not the only benefit of co-evolving together with these organisms. They behave a little bit like a hidden organ, producing vitamins and minerals that are missing from our diet, breaking down toxins, protecting us from disease by killing dangerous microbes on behalf of our immune system, and producing substances that affect the way we smell. Just over the past month, studies have come out claiming that the gut microbiome could alleviate pain from conditions like endometriosis, that it could play a role in preventing cognitive decline, and that by looking at the gut microbiome, we can even predict autism, ADHD, and colorectal cancer progression. Over the past few years, one of the biggest discoveries was finding that the microbiome produces neuroactive compounds, which could be very important in the way we behave. We also know that the success of immunotherapy to treat cancer, one of the next frontiers we'll explore in our episodes, could be linked to the composition of the microbiome. It's like once we started looking at the world through this microbial lens, we couldn't stop finding connections. The tricky part is we don't fully understand what these connections mean yet. Similarly to the discovery of DNA, we suddenly see this invisible coating of life that's been sitting right under our noses. And now we have to figure out how to read it.
So the human microbiome is still largely unknown. Even bacteria, that are the most well-studied types of organisms present in our body, are still largely unknown. We call them the microbial dark matter. The genes that my Escherichia coli is bringing to my body are different from yours, and these are uncharacterized because they are so personal that it's even impossible to some extent to unravel this microbial dark matter.
With the uniqueness of our micropopulations comes the promise of precision, that one day, soon, it could be routine to map out our entire microbiomes, taking a snapshot of all the bacteria living inside of us at any given moment. From there, we could figure out if there's something off, and we could fix it with some tweaks to our diet, a supplement here and there, or maybe some kind of microbial transplant. A recent paper published in Nature, one of the most authoritative scientific journals in the world, reviewed all the ways in which research on the microbiome could help make medicine more precise, and claimed that targeted microbiome elimination promises to be able to remove the harmful microorganisms from the body without disturbing the beneficial ones, by using viruses that eat up bacteria. The thing is, mapping is not the issue anymore, but fulfilling the promise might take longer than expected.
Right now, microbiome tests are cheap enough. We can do accurate sequencing for less than 100 euros. So I think we have everything so that in the next few years we have enough data to cope with the huge diversity and variability of the microbiome to really try to start doing, I wouldn't say maybe personalized nutrition, but at least precision nutrition.
But yeah, so just getting a genome sequence will tell you the species perhaps that are present there. That might be interesting. But does it tell you if you're healthy or not? Well, no, because we don't actually have a baseline for what healthy is. And there's a term that is used in the microbiome literature. It's called dysbiosis. And I hate that term with a passion. The reason I hate it is because people use it as if they know what dysbiosis is. It basically means there's an imbalance in your microbiome. But there's no way to define it because we don't actually know what balance is yet.
We don't know what a healthy microbiome is supposed to look like. But we are starting to see what it looks like when something's off.
We found that people with colorectal cancer, even before starting the therapy or even before knowing to have cancer, they have very specific gut microbiome. At the point that we are developing strategies to do non-invasive screening initiatives based on the microbiome to diagnose colorectal cancer, or at least to suggest the individual to do colonoscopy, which will answer the question with much higher precision, of course, but is an invasive test.
To figure out what it means to have a healthy microbiome, we might need to turn to the gut microbiomes of people who've been living the same way for thousands of years. People who haven't, for the lack of a better term, westernized their lifestyles and diets. These communities, untouched by ultra-processed foods, antibiotics, and modern pollutants, might offer a glimpse into whether we've not only lost biodiversity outside of us, but inside as well.
And of course, in non-westernized microbiomes, we find, for example, in the gut, species that are able to degrade complex vegetable fibers, for example. And when we encountered this for the first time, we asked ourselves whether it's really diet, lifestyle, or is just geography that is responsible for that. And we had the answer from a very old guy, which is 5.3 thousand years old. It's called the Iceman. Was found close to where we are here in Trento, so close to the border with Austria, between Italy and Austria. And this mummy is still very well conserved, 5.3 thousand years old. And by collecting the samples from their gut, we found the microbiome, the ancient microbiome, which was almost identical to that in the non-westernized populations. This individual was living here in the same region, so it's not geography, but he had a completely different microbiome. So we don't eat complex vegetables and we don't need them. But we have to think that evolution put those microbes in our gut during a process that lasted hundreds of thousands of years. So there are a lot of implications about for this loss of diversity, loss of or change of microbial composition in our human microbiome.
And so trying to understand whether these functions actually are important in, let's say, moderating the development of diabetes or something like that is a really leading area of research in the field.
Studying the microbiomes of hunter-gatherer populations could also be interesting for the development of precision diets.
Because if you look at these hunter-gatherer populations, they have some of the most diverse microbiomes on Earth, on the planet. Their diets they're very varied, but they're not as varied as ours. I don't know about you, but I can walk up the street and there's several different, varieties of food that I can get, from Italian food to Indian food to, anything that I want to buy, I can, essentially. So the variety of substrates that I can expose my microbiome to is quite staggering. And if you look at someone in the Amazon jungle, you might think that they have a huge variety of foods that they can eat as well. But the reality is that their diets are pretty consistent. They are seasonal. So there's a rainy season and a wet season, and things change a little bit there. But really the biggest difference is they eat way more fiber than we do. I think that as long as you get fruits and vegetables, as long as those fruits and vegetables agree with you, they don't make you sick or they don't give you diarrhea or anything like that, then I don't see the problem with just eating a smaller number of fruits and vegetables consistently. I just think that that's the key. You need that fiber. And, you also need to sort of understand which are the microbes or which are the food substrates that are best tuned to you.
That's perhaps because our microbiomes form when we're very young and remain pretty stable throughout our lives. Think of a baby's body as fresh, uncharted land, just waiting for microbes to colonize it. It takes 1 to 3 years for this process to finish, and after that, our microbiome settles into a sort of stability. Sure, it fluctuates day to day, meal to meal, encounter to encounter. But for the most part, it sticks with us for life. And here's the kicker. Different strains thrive on different diets. Some are great at digesting plant fibers, but others prefer fats. So when you choose what to eat, you're also choosing which bacteria get the upper hand. Some microbes even communicate with our nervous systems, using dopamine to budget us toward the food they need, almost like training us to make their favorite choices. One way precision diets might work is by charting the microbes in our guts and then advising on what foods best feed them, or suggesting new strains we might be missing that could boost our health. It's like matchmaking, but for bacteria. In Western societies, boosts in our microbiomes often get simplified to just take in probiotics. The term probiotic means for life, in stark contrast to antibiotics, which kill off microbes. Remember Mechnikov from the beginning of this episode, the guy who tried to kill himself twice for science? Well, he was among the first to champion the idea that we could supplement our guts with good microbes to get better at fighting off bad ones. It turned out that most probiotic microbes actually don't stick around in the gut for long. As science writer Ed Yong cleverly put it, commercial probiotics are like a breeze that blows through 2 open windows. That breeze can still help in some specific cases. It can reduce infectious diarrhea or help premature infants avoid a horrible gut disease called necrotizing enterocolitis. But to truly impact our microbiomes, we would need more tailored probiotics.
Probiotics are not the things that you buy from the health food store for the most part. So I'm even seeing the term, probiotic yogurt and probiotic this, and to me that's it's a shame because it undermines the good work of the people have done on seriously clinically proven probiotics. That takes a lot of effort and time and money to get a proper probiotic through clinical trials and onto market. So if you are saying that you're going to have to make a personalized, treatment regimen or personalized strain or different groups of microbes for different people, each different group of microbes, each different drug that you're producing has to undergo testing. And that testing costs thousands and thousands of dollars. And so it very, very quickly becomes not financially viable or not economically viable to develop personalized medicine, except for perhaps things like cancer treatment.
Even if precise probiotics never make it to the market, and we'll see about that, we should be equally wary about what boosts our microbiome and what damages it. Our lives are filled with habits and substances that can damage the microbes in our bodies. Take antibiotics, for example.
And the thing about antibiotics is that they have very broad-ranging effects for the most part. And so an antibiotic that might kill, a nasty strain of, let's say, E. coli or salmonella or something like that, will probably also have a major effect on other microorganisms. The microbiome is damaged by antibiotic use. But for the most part, when a doctor prescribes an antibiotic to you to treat an infection, he or she has done that risk-benefit equation and shown that, if really you're much better off taking the antibiotic to get rid of the infection, and we'll worry about the microbiome damage. Later maybe the microbiome damage is a lesser problem than the actual infection.
So antibiotics are clearly impacting the human microbiome. But I don't think antibiotics are the drugs that are most dangerous for our microbiome. This is basically because antibiotics are taken for 1 week, 2 weeks, but they are not taken for a very long period of time. There are instead drugs taken for decades and are changing the pH of the stomach and also of the gastrointestinal tract. And this means that what we see is...
This isn't to say we shouldn't take medicine when we're sick. Of course we should. But let's leave it to the doctors to decide when we need those drugs, instead of playing pharmacists ourselves.
But essentially, we're the zookeepers, right? So we don't want to let our animals die. So how do we not let them die? Well, we look after them. We provide them with the best possible food and the best possible attention. And so
today, microbiome research runs into the same challenges as DNA studies. At first, it was celebrated as a discovery that would change everything. And in some ways, it really did change fundamentally the way we understand our bodies. But there's a real risk that if it doesn't provide quick answers to all of our questions, it could end up being dismissed and pass as just another overhyped idea that didn't quite deliver. And we'll all move on to the next big thing.
And I think that, investors, I think that just the population in general is kind of lose patience. They don't realize how long it can take sometimes for research to take place and for us to do it properly. The pressure is always on. I don't think that we've given it a fair shake. I don't think that we should give up. Absolutely not. But I also think that, unfortunately, in order to move ahead in the research field, there needs to be money. Neither
DNA nor the microbiome will ever fully explain what makes us sick or keeps us healthy. But this doesn't mean researching these fields won't be life-altering for many current and future humans. In the first episode, we explored how our health and our responses to treatments depend on DNA, which serves as a guide for building every cell in every living organism. Now we've seen one reason why DNA alone can't tell the whole story. We're living beings surrounded and inhabited by other living beings. In the next episode, we'll dive into how these first two unique maps interact with other layers that shape our individuality, and that, in a fairly near future, could lead to ever more precise diagnoses and treatments. The Science of You is a Cora Media podcast, supported by the European Partnership for Personalized Medicine, coordinated by Fondazione Toscana Life Sciences, and co-funded by the European Union's Horizon Europe Research and Innovation Programme. The podcast is written and hosted by me, Silvia Lazzaris, with editorial support from Simone Clemente and editorial curation by Graziano Nani. Sound and music supervision is by Luca Micheli. Post-production and editing are done by Andrea Girelli. The sound engineer is Luca Possi. The project manager is Silvia Arcidiacono Mutti. The producer is Martina Conte, with post-production coordination by Matteo Scelsa. Thanks to Francesco Zaffarano for his voiceover.